An inside look at the strange world of genetic engineering on Animal Farm, next.
The following program contains occasional disturbing scenes. Parental guidance is recommended.
If you could grow a nose in a jar, would you do it? If you could grow yourself a new heart inside a sheep, would that be right? If you could modify your DNA to allow you to grow back lost limbs, would that be going too far? So far in this series, we've dined on super-sized cows, met rabbits that glow in the dark, and come face to face with featherless chickens. In this episode, we discover how we are creating new animals and plants that could save our lives. This is not tomorrow's world. It's happening now. If all the creatures created by science were gathered together on one farm, then this is what it would look like. The farm is imaginary, but all the plants and animals you are about to see are real. Welcome to our Animal Farm. In this episode, our tour of Animal Farm exposes an incredible new world of medicine. We've placed 2 people on the farm to help guide us around. Olivia wants to persuade you of the benefits of genetic engineering and biotechnology.
I'm Olivia Judson, and as a biologist, I believe that biotechnology is a tool with amazing potential power. By using techniques like genetic modification or tissue engineering, we have the potential to create crops that could help us to control infectious diseases, or animals with human organs growing inside them. But in order to really take advantage of the technology, we're going to have to embrace it.
But organic food fan Giles Corran finds the idea of a pharmaceutical farm highly disturbing.
As a food writer, I really believe that farms ought to be left to do what they've always traditionally done best, which is to provide food. I really don't like the idea of their being turned into factories to produce drugs and human organs.
Olivia Judson is an expert in natural selection and evolution. But she is now going to step into an extraordinary new world. A world where scientists have taken control and turned nature upside down. Inside this barn is a herd of cows that evolution could never have produced.
What a sight! Every animal here is a clone. They're all genetic copies of just 2 original cows created in the lab. They were created by genetic engineering in the hope of saving human life.
Hey!
These calves are the brainchild of geneticist Professor Jim Roble. He's built them from the ground up to help cure some of the most dangerous diseases on the planet.
So what is she? What makes her special?
Well, this is a calf that has a series of gene knockouts, and she also carries the human microchromosome that has the human antibody genes.
So she can make human antibodies in her blood?
Yes.
Antibodies are produced by our bodies to fight disease. They can destroy lethal infections like SARS and poisons like botulism. But no one has found a way to mass produce them until now. Dr. Roble's calves are worth up to $500,000 each. They have human DNA inside them. Their future is to be milked for their blood. This is the world's first antibody extraction parlor. For 2 hours every week or so, these precious cows are connected to pumps that filter their blood and extract human antibodies.
Making antibodies of this sort on a large scale has long been a dream of medical science. They could be used to fight diseases like anthrax or Ebola, for which there is no known cure.
if we want to make antibodies to treat a specific disease, what we'd really like to do is to inoculate you over and over again so that you develop a very high titer of antibody against that agent. But we can't do that. We can't treat people that way.
So if we wanted to make antibodies against anthrax, we can't, because we can't go around infecting people with anthrax.
That's correct, yes.
So instead of humans, Dr. Robles decided to use cows to create his antibodies. Long-lived, and with 8 times our blood supply, he wants to turn them into reliable, high-volume antibody production lines. The ultimate goal is to, say, take anthrax,
Yes.
expose the cow to anthrax, inject it with anthrax, at which point it starts making lots of antibodies against anthrax.
Yes.
And then you can collect those antibodies, purify them,
That's correct.
and treat people who have been exposed to anthrax.
Yes.
But it's no easy job to get cows to produce human antibodies. You have to genetically modify the cow's immune system to make it at least partly human. Dr. Roble has had to copy 2 sets of genes out of our DNA, which he then combines into a package called a microchromosome. But to get that microchromosome into the cow, he's had to use one of the most complex cut and paste jobs I've ever heard of.
Take the microchromosome and you transfer it from mouse cells to chicken cells. Okay? And this is a specific chicken cell line that allows you to cut and paste chromosomes. And the best way to carry this chromosome is actually in a hamster cell line. So then it goes to a hamster cell line. We then transfer it to the cow cell line.
So let me just get this straight. To make a human chromosome, you have to go through mice, chickens, hamsters, and then cows.
And then finally cows, yes.
Wow.
Because each cell type has its own peculiar but specific quality that allows us to do a specific kind of manipulation.
And this means that they will make human antibodies in their blood.
That's correct.
These cows could be as important as the discovery of vaccination or antibiotics. They could be used to defeat diseases that no antibiotic can cure, maybe even cancers. They could be an entirely new weapon in our fight against disease.
And how much can you take from a cow before it starts to feel uncomfortable?
A cow like this, you could easily collect probably 10 liters. A cow has about 20 grams of antibody per liter of plasma. Then you're talking about 200 grams of antibody.
These cows take blood donation to a whole new level. Just 10 or 20 of them could supply the yearly worldwide demand for one of Dr. Roble's antibody therapies. And it's the kind of advance that only genetic engineering could have given us.
Dr. Roble's cows have been created to make human antibodies outside a human being. But the science doesn't have to stop there. Could we ever grow a human organ independently of a body? For our next stop, we're going to meet the farm's tissue engineers, whose job it is to find out.
My face, bog standard. 1 mouth, 1 nose, 2 ears, 2 eyes. Just the way I like it. But down in the basement, they're up to something called tissue engineering.
Tissue engineering is the science of growing organs or tissues in a laboratory. Here on the farm, the process begins with a mould in the shape of Giles's nose.
How do I look? Better or worse?
The mould is used to make a structure called a scaffold, on which cells can grow.
And that scaffold's got lots of special properties that are going to allow cells, the cartilage cells and skin cells, to actually form the living part of your nose structure. And we actually start with a plastic material, which is this material here, actually used to make biodegradable plastic bags in supermarkets these days, but this is a much finer quality. So that goes into the oven.
To make a solid structure, the scaffold is heated for about an hour.
Right then, let's have a look.
Okay.
My, doesn't that look yummy? That's pain.
So that is the scaffold structure.
My word, that was my nose.
That's right. 90% of that is actually air. It's only 10% that's been taken up by the polymer structure. And that 90% is giving the space for the cells to get into there later on in the process and form the tissue of your nose.
That's really rather impressive. I can't wait to see what happens next.
We'll leave Giles's nose to stand on the sideboard for an hour or so. It's time for Olivia to see some of the most exciting work on our farm. What will she make of the farm's baby department? Here they want to grow a human liver.
It's a boy.
This is Dr. Nico Ferraz. His first job of the morning is to collect the umbilical cord of a newly born baby. He's looking for an ingredient that is revolutionizing modern medicine.
20 years ago, umbilical cords like this were usually thrown away. We didn't know that they were one of the best places to get hold of stem cells.
Here's the cord, a very long one.
So this is the umbilical cord?
This is the umbilical cord.
So the idea is to collect the blood and then extract the stem cells?
Yes, absolutely.
Why do we care about stem cells?
Well, stem cells are really exciting because they have the possibility to build many cell types of the body. It just opens huge possibilities.
So what are stem cells? They are all originally made from stem cells. As we grow in the womb, these stem cells develop into the tissues that make up every part of our body: bones, muscles, brain, skin. The point about stem cells, the reason everyone is so excited about them, is that they have the power to become any part of the body. Stem cell therapies are already saving lives on the farm. Olivia's now going to visit one of the farm's resident humans, baby Ross.
What sort of future would he have been facing?
He wouldn't have had a future, basically. He just wouldn't. Basically, SCID babies don't live past a year old.
SCID stands for Severe Combined Immune Deficiency. Ross's body couldn't produce a particular type of cell called a T cell that helps the body fight off even the smallest infections. But doctors transplanted stem cells into his bloodstream, where they multiplied and transformed into the T cells that Ross needed.
He wasn't able to fight off any infections, so basically he would have been dead by a year.
So even a common cold could have killed him?
That's correct, yeah, or a kiss or anything. So for the last 6 months we haven't kissed him at all. Aww. He hasn't been able to touch our hand, touch our faces, touch anything. He do what he wants, I don't care.
Stem cell therapy saved Ross's life. But there's been a lot of controversy over stem cells, because one of the places we can get them is from embryos. However, embryos aren't the only source. We can also get them from bone marrow or umbilical cords, as Nico does.
Nico's dream is to grow a human liver from the stem cells he collected earlier. Growing a human organ from scratch? One of the most complex parts of the human body? I wonder how he's getting on.
So you want to make a little piece of liver tissue that will can actually work as a liver?
Absolutely. In the long term, we can envisage maybe creating enough liver tissue to assist a patient with liver disease or liver toxic crisis. The difference between life and death in a patient with liver conditions is it's only 5%. So 5% improvement can keep them alive for a decent amount of time.
It looks like step 1 to growing your own liver is to crack out something Nico calls an anti-gravity machine.
So this unassuming package is the bioreactor?
That's right.
This is the anti-gravity machine?
That's correct. Impressive, isn't it?
The bioreactor was designed by NASA to reproduce the effects of being in space. It reduces gravity, allowing stem cells to grow in all 3 dimensions.
A bioreactor is actually mimicking microgravity. when you create tissue in 3 dimensions, you get a lot of forces and physical stress on the cells that you are actually building in 3D. So this machine suppresses those nasty effects on the cells and enable us to grow liver tissue in 3D from cord blood stem cell.
Step 2 in growing your own organ is to load up the bioreactor with stem cells. Now Nico's got to cajole them into deciding that they want to become liver.
So basically you put them into this sort of anti-gravity machine, and then you add various compounds and chemicals that say become liver, become liver, become liver.
It's got all the salt, minerals, sugar, proteins that they need to be alive, and some special reagent called growth factors.
Growth factors are Nico's magic ingredient. They convince the stem cells to start growing and also tell them what to turn into. In this case, liver.
So this is kind of stem cell medicine.
Stem cell medicine, stem cell soup to make them grow. So I'm just going to put this on rotation in this hot chamber.
All right, let's...
Stem cells, some pretty potent chemicals, and a bioreactor. And Nico could have a production line for human organs.
Oh yes, it's very warm.
Nice and warm, yeah. So it keeps them happy at the...
Comfortable cells.
Comfortable cells.
After 6 weeks, this is the end result, a 3-dimensional chunk of working human liver. It may measure a few millimeters across, but this could be the future for organ transplants. Just a few grams of this tissue could save the life of someone dying from liver failure.
We haven't finished with the organ farmers yet, but we need to get back to the nose kitchen, where Kevin, the tissue engineer, is firing up a pressure cooker, the likes of which you've never seen before.
So far, all Kevin's done is bake a nose-shaped scaffold, but now it's time to start cranking up the serious technology. Kevin's going to show me how to make my nose come alive. Right. This is what? This is an extraordinary piece of equipment. This is exactly like I imagine that science laboratories ought to be.
Yes.
You just whack the nose in there, turn it on, and it creates.
Exactly, yeah. It's actually a controlled chemical reaction that's going to make this into a scaffold that attracts the cells to it.
So it's going to change the nature of the nose scaffold?
Yeah, you won't actually see any difference to the eye, but actually a lot of important changes are happening to the chemistry of this material.
Wow. So you put it in there and it changes it into something which will fool the cells into thinking it's a living thing?
Yes, that's right, yeah. Exactly.
Cunning.
And so it just goes through this front port here, and then we just position it.
Frankenstein brought his gnarly old monster to life using a lightning conductor. Just imagine what he could have done with one of these.
You can see now within the chamber it's glowing pink, and the reason for that is the gas that we're leaking in there is being charged up, so it becomes very reactive, and that will now undergo a chemical reaction with your nose scaffold and change its surface.
Kevin's cooker is turning Giles's nose into a magnet for living tissue. Almost literally. Through a complex series of chemical reactions, this machine is making his nose positively charged. Any cells that come close to it will stick like magnets onto the scaffold surface.
Okay, well the time's up now and the chemistry of the system will have been changed, so I can now take the nose out for you. At this stage I've got gloves on because we don't want to interfere with that chemical process. Now your nose doesn't look any different, it still looks like a polymer scaffold of your nose, but actually to the cell it will look very different. It will now have a different type of surface which the cell will want to stick to.
So what do we do with it now?
Well, it's ready for the cells now, the next step.
Now Kevin's really cooking. He's doused my nose in meths to sterilise it. given it a quick wash, and then it gets its growth factors, the primers for life.
It's basically got all of the salts and some of the nutrients that your cells need to form the tissue. The nose is now ready to receive the cells.
But it's quite rough still, the nose. I mean, is it going to make a rough flesh nose, or is it going to be smooth like mine?
Over a period of time, the polymer structure, which is causing it to be rough, degrades away. It's deliberately rough at the moment because that's giving it the pores that you need to actually allow the cells to get in there. So what I'm going to do now, I'm just going to put this pipette onto here, and I'm going to now collect some of the cells, probably around about 10 million for a nose of your size.
How many for a nose of Charles de Gaulle's size? Many more millions?
Many more millions.
So now Kevin's basting my nose with a pink potion of countless living cells. There's now millions of human flesh cells around the scaffold of my nose.
Yeah, and from the earlier process where we changed the surface chemistry of it, they should now be finding that an attractive surface to stick to.
Let's give the cells time to get used to their new home. Harvesting noses and livers in the lab is one way of making spare body parts. But here on the farm, some scientists have an even more radical approach. What would happen if we could grow human cells inside an animal? Olivia's about to find out. These sheep are up to 15% human. That's right. Bioengineers have worked out how to grow an animal that is part sheep and part human being. It all starts in our operating theatre. Here, scientists are going to extract a sheep embryo from its mother's womb, inject it with human stem cells, and then sew it back in. The lamb, when born, will contain sheep and human cells. It will have become humanized. Professor Zanjani has spent 30 years on this project.
So what is a humanized organ?
A humanized organ would be an organ in which at least part of the organ is derived from human cells.
Dr. Zanjani's humanized sheep are made to order. Each one is precisely matched to a donor, a patient who provided the original stem cells.
We would go in and take about a couple of ounces of bone marrow cells from that patient. We isolate the stem cells from them, transplant them into the sheep fetuses, and then within 2 months we will have a liver that's at least 10 to 15% made up of the liver cells derived from that patient.
I can't believe this is going to work. Professor Zanjani says he can simply inject human stem cells into a sheep, where they'll survive, multiply, and turn into human organ tissues.
We inject the cells into peritoneum of these animals, and then these cells get distributed through the lymphatic system into the circulatory system and get distributed to all the organs in the body.
The human and sheep cells live quite happily alongside each other inside the sheep. But there is one risk to Dr. Zanjani's process. If DNA from the sheep cells and the human stem cells gets mixed together, we could end up with what is called cellular fusion. And some people are concerned that this could produce a strange new hybrid that has the features and characteristics of both sheep and humans. It is a possibility Dr. Zanjani has considered.
In this model, transplanting the cells into the fetal sheep at this early stage does not result in fusion at all. We have not seen one single fused cells. So we either have sheep cells or human hepatocytes or liver cells.
So they maintain their identity as sheep?
They're regular sheep, they behave like sheep, they don't speak like humans, they don't behave like humans, and they don't feel like humans. That's very important.
Right. I want to see how on earth this works. The first thing they do is cut open the sheep's stomach.
It's like the unnerving smell of burning flesh. Are we going to to take it all the way out, the uterus, the whole uterus. And they're a different shape from a human uterus, aren't they?
Yes, they are. I've seen some weird stuff in my life, but this is something else.
Looks a little bit slippery.
That's the sheep's womb being pulled out, with a living lamb fetus inside it.
Oh my God, it's enormous. Oh, wow.
Now comes the critical moment. The surgeon has to feel the living fetus so that she can identify the area into which the stem cells will be injected.
Inside this horn. So I just gentle palpate. Now I'm feeling where the ribs are. They are right here.
The stem cells must be injected into the peritoneum, the stomach area of the fetus.
So basically, when a human stem cell gets to the sheep's brain, it will become a neuron.
Yes.
And when it gets to the sheep's liver, it will become a liver cell.
A liver cell.
As the human stem cells travel around the sheep embryo, they grow and transform themselves according to where they settle. The sheep's liver, heart, lungs, possibly even its brain, will all become partly human. Ultimately, Dr. Zanjani hopes to farm hundreds, even thousands of sheep to produce organs for transplant.
The nice part of all of this is that the 2 ounces of stem cell or bone marrow cells you get from these patients provide enough stem cells to do maybe about 10 fetuses.
Wow.
So it isn't just you have one organ for transplant purposes. You can have many of those animals are still available in case the first one you give fails and so on.
What an astonishing idea! That one day we all might have a personal flock of sheep on standby to provide us with new organs if any of ours fail.
Dr. Zanjani's work is still in the developmental stages, and receiving a transplant from his sheep isn't possible just yet. But some organs created outside the body are being used in people right here and now. For our next stop, we're going to meet someone whose life has been changed forever by biotechnology. Caitlin McNamara was born with spina bifida. Her bladder didn't work, so she received the world's first biotech bladder. Caitlin's bladder was grown from scratch in the laboratory.
So, first of all, you had this operation how long? 6 years ago now?
Yeah, February of 2001, I had the cells harvested.
It's just what they did with my nose. They built a scaffold, they got some cells, and hey presto, Caitlin's new bladder was growing. So they made it on a mould and then put it into you alongside?
Yeah, so that it makes it bigger.
It's clear this technology has changed Caitlin's life. With spina bifida, the simplest things were a challenge. But her new bladder has made it possible for her to do the things most teenagers take for granted.
I guess.
So you're like the first person in the world to have this kind of surgery. How does that make you feel?
Happy, because it's been working for 7 years now.
So there are lots of things you can do now that you couldn't do before, I guess?
Yep. I can do whatever I want without having to worry about having accidents or having to go to the bathroom, like really, really fast.
So that's going to last you for a lifetime, the bladder, or do you think you might have to do it again?
I think it's going to last me a long time. I don't know exactly how.
And if not, they can grow another one, I guess.
Biotechnology can offer powerful solutions for individuals like Caitlin. But could it ever be used to tackle disease on a global scale? For our next appointment, we're heading to the greenhouses, where the most lethal plant on the planet is being transformed into a lifesaver.
Tobacco, most people associate it with cancer, and you want to make it into a plant that is going to potentially save the lives of millions.
Yeah, well, I think most people are very familiar with the concept of getting medicines from plants. We've done it for hundreds of years. But with the advent of new technology, particularly genetic modification of plants, we can take plant species like tobacco and turn them into factories for making new medicines.
This is great. Dr. Ma wants to take tobacco, that killer weed, and use it to help stop the spread of a disease that's killing millions, HIV/AIDS.
HIV is a huge problem, of course, around the world, but particularly in developing countries, where there are millions of people who need this kind of protection, but who essentially can't afford to pay for expensive modern medicines.
So explain to me how it actually works.
Well, we're looking at production of something called an HIV microbicide. So this is a compound which inactivates the virus when it comes into contact with it.
Dr. Ma has found a way to kill HIV. A compound called cyanovirin that destroys the AIDS virus on contact. He wants to turn cyanovirin into a cream that can be used as a barrier against HIV infection. Something women could use to protect themselves without worrying about condoms. But the only thing that produces cyanovirin naturally is a tiny blue-green algae. And that's not much good if you're trying to make a cream for millions. So Dr. Ma has taken the gene that produces cyanovirin from the algae and put it into 10-foot-tall tobacco plants.
So these are the plants?
Yeah, so once we've established the little plant that's downstairs, they come up and we pop them into soil. And now it's all about letting them grow, generate biomass, and also to let them go to flower and eventually go to seed, because that's where the scalability comes in. One plant will probably produce about, 10,000 seeds.
This is a fantastic use of biotechnology. Once Dr. Ma has created one plant that makes cyanovirin, his dream is to take thousands of seeds from it and ship them to different countries. There, local farmers could cultivate acres of these plants, allowing his HIV barrier cream to be mass-produced around the world.
Plants are cheap to grow. Once you get to this stage, you're actually in very low-tech technology. Anybody can build a greenhouse very cheaply, even a contained greenhouse, and grow a number of plants. And if you really want to address global access to modern medicines, you have to think about new technologies, and that's where I think plants come in.
Just a single leaf on a single plant in one corner of the farm. But the work of scientists like Dr. Ma could ultimately end up saving the lives of millions.
Down in the basement, the tissue engineers are still hard at work on Giles's nose. It's been bathing in a mixture of human cells and growth agents for 3 weeks now.
It looks horrible. What have you done to my nose? That's years of heavy drinking. You wouldn't think that could do that in 3 weeks. It's still recognisable just as my nose, but it's my nose in 30 or 40 years' time if I don't look after myself.
The cells have now multiplied within here, filled the space and laid down some cartilage.
Cool. Wicked. So what do you do next?
Well, the next stage in the process is to put some skin over the top.
In a way, you could have just taken some skin from my arse or something and stuck it on the nose.
Yeah.
But as we thought that was not going to be very comfortable, we can just, it's an alternative to a skin graft, but it's
exactly, yeah.
Cells you've grown.
These days, scientists are able to produce skin grown to order. All they need is a few cells, and they can make as much or as little as you want.
You see it folds back up, back open.
And you just float it onto it through the solution?
That's correct.
It's really rather impressive, actually. It looks almost alive. It is alive, I guess. The cells have populated the scaffold, multiplied, and now skin is going to start growing too. Wow. Ugly, but amazing. So that is now the scaffold of my nose with some cartilage in it and then proper skin on the outside?
That's correct.
Cool. But of course, Kevin doesn't have to stop here. He's got a whole crop bubbling away in the incubator, all in various different stages of growth. But what's he going to do with an oven full of noses? And that's not the ultimate colour of nose you're aiming for?
No, the ultimate colour really is determined by getting blood vessels into it. So although we're not clinically interested in growing noses, but when you are growing a tissue, you need to get blood vessels into there.
Why are you not clinically interested in growing noses?
Because there's some really big challenges out there where patients need new bone or cartilage for joints to be grown. And that's really where I think this science can make the biggest impact, and is making the biggest impact.
Right. So in fact, it turns out noses are the easy bit. Growing a nose is a good way of getting to grips with the basics of tissue engineering. But this is really just a launch pad for the real medical innovations.
And I think they're looking now at using tissue engineering to add to their tools of techniques.
But you can make more exciting things than noses, you're saying?
It depends who you are, but I think if you're a patient who had a major break of a bone that wasn't re-healing, what would be exciting to you would be to regenerate that bone. Or if you're a patient who's lost...
Is that what this technology has been developed for, then?
Really, yeah, to look at the bone and cartilage associated with joints and to get people with an improved lifestyle, because the tissue actually regenerates within their body.
The hope is you could be seeing a tissue engineer like Kevin in every hospital. His skills could heal burns and repair broken bones more completely than any surgeon.
The alternative therapies are very poor, and this really offers an opportunity for the first time for man to actually regenerate and cure those patients.
So it's quite possible that from the tiny acorn that is my nose, a mighty oak tree of medical innovation will grow.
We're nearing the end of our tour of the farm. We've learnt how to make cows with miracle blood, sheep with humanized organs, even grow a nose in a jar. But it doesn't stop there. What if we didn't need to grow organs in a lab, or even in a sheep? What if we could chop off our fingers or arms, and then they would grow right back? This work has already begun on the farm.
I've been told that what could be the holy grail of medicine has been discovered by pure accident in a routine mouse tagging exercise. I suppose it's just the kind of accident you'd expect to happen in a lab.
We had gotten a group of these mice. We ear punched them with different hole codes.
Hole punch,
a little hole punch. And 3 weeks later, when I came up to look at the mice to see if there was any effect of the experiment that we were doing, the ear holes had absolutely disappeared.
During the course of a routine experiment, Dr. Heber Katz unexpectedly discovered that these mice have a truly remarkable ability.
We were beginning to be very excited, but we had to redo it. So we ear punched the mice again, and then we watched them for 3 weeks, and sure enough, the holes got smaller and smaller and disappeared. This mouse was actually ear punched 2 days ago. I will show you a mouse that was ear punched 11 days ago.
If you look closely, you'll see there's no scarring on the mouse's ear.
And this mouse was done 3 weeks ago.
In fact, these mouse ears aren't healing in any conventional sense. They're regenerating, as if the injury had never happened. This ability to regenerate is an extraordinary power. It can be seen even more dramatically in a few other animals. If you cut the arm off a starfish, or the leg off an amphibian like a salamander, it just grows back. But it was assumed that over millions of years of evolution, mammals like humans and mice had lost this power. Until Dr. Heber Katz discovered her mutant mice. They come from a strain of mice often used in scientific research. By complete accident, when these mice were being created, their regenerative abilities were switched back on.
When I saw the pictures of what was happening in amphibians, I realized that this was, very similar to what was happening. And I actually spoke to people about it, and I said, well, what about mammals? They said, well, mammals don't regenerate.
And if Dr. Heber Katz's mice can regenerate, it's not impossible that other mammals could regenerate too. We might have the same regeneration genes that have been switched on in these mice. And although they aren't yet growing back entire limbs like a starfish, Dr. Heber-Katz's mice do show some extraordinary powers.
Besides the ears, we've looked at the heart and made injuries to a small portion of the heart, and the heart heals, regenerates, function comes back, the architecture comes back, and they heal almost perfectly.
Once the genes behind this regenerating power have been identified, the consequences for medicine could be enormous.
There are a lot of applications for this. I mean, hopefully we will be able to regenerate tissues, organs in all mammals, in humans.
In humans?
Yes, absolutely.
So you're saying you'll be able to do this in our lifetime? You'll be able to, people will be able to grow back bits and pieces?
Well, I think that we are going to be able to regenerate. I mean, I think that it definitely is a possibility. The other possibility is that we will be able to replace cells all the time, so that we won't have aging populations of cells.
The regenerative mice were created by accident. But elsewhere on the farm, mice with mutated DNA are being deliberately produced by researchers. In the final stop on our tour of creatures being studied are so valuable, he must don protective clothing to ensure he doesn't contaminate them. In this labyrinth, mutant mice are being scrutinized for their genetic secrets. And there are literally thousands of them.
The mouse is a good model for the human, although it's small and furry, and we're big and we're not very furry. We know that its physiology, its biochemistry, the way it works is very similar to humans.
It's like Mouse City. Look at them all in their high-rise blocks. Apparently, mice and humans share over 90% of their genes, and for these geneticists, that's remarkably useful.
We know that in the human population, we're carrying all sorts of alterations in our DNA. You're carrying alterations in your DNA. Those alterations will make you more susceptible in the future to Alzheimer's disease, cardiovascular disease, diabetes as you get older. We need to know the relationship between those alterations and what's happening in the human population. So we can study how genes cause disease in mice, and we can then translate that information into working out how genes cause disease in humans.
To find out how genes work in mice, the animals are first injected with a chemical that randomly alters the DNA in their sperm.
All these male mice here have received an injection, a course of 3 injections in fact, that damages their sperm. So when their sperm actually regenerates, all their offspring should be carrying mutations.
The male mice with the mutated sperm are now placed in cages to mate with females. When the offspring are born, each is carefully inspected to find out what unusual new traits it might be carrying.
He does look quite normal. With the full screen, we will actually check that their teeth are there, they've got the right number of toes, the right number of tails.
Right.
And
do they often have more than one tail?
Not often.
The mutations in the sperm are totally random, so no one can predict what kind of mice they'll create.
She's actually got quite wonky ears there.
Once an unusual feature is discovered, the scientists look for the gene that's causing it. And it seems they're searching for everything, from genes that cause cancer to genes that cause dominant behaviour. But my favourites are the mice with a more specifically human flaw. Right, so these are the alcoholic mice?
Yes, these are the ones that are on the alcohol preference testing.
And they have a preference for it?
Yes, you can see, I don't know if you can see in the cage that there's 2 bottles. One of them is with ordinary water, and one of them with medical grade, well, you can see on there, 3% alcohol.
Which is quite a high 3%. Scientists believe that part of the reason people become alcoholic is genetic. These little fellows are on the equivalent of 2 bottles of whiskey a day.
This is actually a preference test, and the mouse itself has a choice.
Right.
Does he want to go to the water bottle, or does he want to go to the alcohol?
As do alcoholics, frankly.
Yes, yeah.
To find out why the mice drink, they are anaesthetised and placed in a brain scanner. Researchers discovered the animals lack a crucial piece of neurochemistry, which leaves them feeling jittery and irritable all the time. And it's believed that drinking alcohol makes them feel more relaxed. The gene that causes this problem has been discovered, and the impact of the finding could be enormous.
There's no doubt that if we understand more about the relationship between gene and disease, we're not just understanding the symptoms, we really understand the mechanism of what's causing a person to drink too much alcohol.
And this kind of research could lead to a new era of personalised medicine.
In the future, a GP will take a blood sample from you. They will look at all the genes that might be involved in all these diseases, see whether you're carrying alterations in your genes that say you're at risk of disease. And then there's enormous opportunities to change your lifestyle, or perhaps even to intervene with drugs to ensure that we put off the day when you might develop those diseases.
They really have got everything down here. Just round the corner from the alcoholics are mice with an even more topical condition.
Nice to meet you.
Nice to meet you. What have you got here?
We've got some obese mice. So if I get these out, so these are brothers. They're about 14 weeks old. And so this one is about 50 grams, and his brother is about 30 grams.
Gosh, so what with me being 12.5 stone, that is like having a 20 stone brother?
Yes.
I would not hang out with him in public.
These mice have a genetic mutation which means they can't tell when they're full up.
He's actually burrowing into his food up to his... all you can see is his huge arse.
As a result, they just carry on eating.
This place is in Aladdin's cave of understanding, directly relevant to us all. It's incredible to think that because of genetics, future medicine could be tailored to our individual needs.
Manipulating our DNA. As a route that we don't want to go down, and a route that we don't need to go down yet. We know because of the complexity of the genome that messing with it is a dangerous and difficult business. What we want is to understand the relationship between genes and disease so that we can intervene, not at the level of the gene, but at how the gene is affecting the biology of that particular person, and how we can